I'm delighted to be here today to talk to you about this new program on which Steve and I and a group of others have worked very carefully through the year to realize its this new initiative in a way that I think will be terrific for the general cardiovascular research community. And it's entitled Cardiovascular Genome-Phenome Study. GPS obviously has implications for guiding us through the complexity of cardiovascular phenotypes. And to set the stage, let me remind you of the complexity of the universe within which genomics and phenomics sit. Here we have across the upper part of the panel the genome through the proteome and metabolome, ultimately leading to the phenome or the pathophenome. Underlying this sequence are the transcriptomes, epigenomes, and metagenomes that interact with, modulate, alter the expression of genes that are coded for in the genome. And beneath this, we also have other kinds of environmental factors, including therapeutic perturbations, environmental exposures, which modify the epigenome as well as the proteome, post-translationally, for example. So this constellation of stages of transition from the simple, if you will, simple linear expression of the genetic sequence that we all carry in our 3 billion base pairs through to what we are, both in health and disease, is what sets the stage for what CVGPS is all about. We can simplify that set of transitions as I show here, where the genome interacting with and modified by the environome, what we eat, what we breathe, and the bugs that live within us commensally, all conspire to create a unique individual, again, in health and disease. So we believe, the American Heart Association believes, that a combination of rigorous population studies and molecular analyses using cutting-edge genomics can help define more sharply subgroups of individuals who are either at risk for a disease or have a disease, and it is that subgrouping which can facilitate our development of precision medicine approaches to personalized treatments, diagnostics, and prognostics. The AHA is uniquely positioned in CVGPS as a catalyst, really. It is an honest broker that facilitates the utilization, analysis, and distribution of pre-existing data sets, such as those that exist at Framingham and Jackson. These well-established cohort studies that have provided so much useful information in our understanding of the factors that determine the risk of cardiovascular disease have, in the modern era, also collected a great deal of data that would be viewed as an important element of the genomic structure that we wish to put into this CVGPS platform. The AHA, working with the partnering institutions that facilitate and lead the Framingham and Jackson Heart studies through contract, Boston University, University of Mississippi, Jackson State, and Tougaloo College, have all worked together to allow us to present to you what ultimately is this growing integrated population study platform that will also expand further with the addition of other cohorts and study populations to provide a truly fully integrated platform for cardiovascular genomics and phenomics. The overall program structure is shown here. We have a volunteer oversight committee on which Steve and I and others sit. There's ultimately a virtual cardiovascular database and biorepository, which we hope to build as part of this program that the AHA will help support. The AHA will also seek venture funds and industry collaborations to provide additional levels of true financial support as well as support in kind. And the ultimate goal is to work with our partnering institutions, which I mentioned on the last slide, to provide deep phenotyping, orthogonal phenotyping, integrated phenotyping, along with all levels of genomic analysis with the appropriate analytics. And I can't overemphasize the importance of the analytical portion here. And even though these data are extraordinarily rich, they're nothing without the appropriate analytical strategy. That these analytics will provide the kind of knowledge and insight that we can gain optimally from this integrated genome and phenome data set. Education is also a crucial element to the whole process since these are technically complicated kinds of analyses and kinds of processing that need to be more widely disseminated with standards developed for our trainees to acquire. The specific programs we've talked about fall really into 4 broad categories. The deep phenotyping, which would include a phenorepository, time-dependent monitoring of phenotype, orthogonal phenotyping, that is, features of phenotype which may not bear directly on the disease of interest or the disease risk of interest, but may actually have an important bearing on that disease or disease risk once the molecular determinants of those orthogonal features are apparent. On the genomic side, we have whole genome and whole exome sequencing, gene expression, proteomics, metabolomics, and time-dependent monitoring of these genomic data sets. The kinds of analytics, as I mentioned earlier, are becoming increasingly complex, but also increasingly important as the levels of complexity of the data sets we gather increase. And those include network analyses, systems analyses, the Health eHeart study from the West Coast, which provides an additional level of phenotypic analysis, and then the educational features required for genomic as well as bioethics considerations. Research supporting grant offerings that we've put together for this program really fall into 3 categories at this point in time. There is support for infrastructure, sample handling for data and biosample requests at Jackson and Framingham, the development of an integrated database, as I mentioned, perhaps metabolomics core with dedicated MS and MS analytics. We've recently, we are in the process of planning a grand challenge RFA, which would be $500,000 a year for 4 years. It needs to be interinstitutional. It needs to be interdisciplinary. It should utilize Jackson and Framingham resources and/or other study populations established or proposed. And it should focus on hypothesis-driven topics some of which we will include as possible considerations in the RFA, such as determinants of longevity, neuroomics topics, racial disparities. What we've just released, and what you'll hear from Steve later on today about those grantees who have been announced recently, are the first pathway grants, and these are $250,000 per year for 2 years. They may cycle as an RFA as follows: 1 per year investigator-initiated and 1 per year targeted. They will utilize Jackson or Framingham resources and/or other population studies, either established or proposed, and they should focus on potential broad topics, including phenotypic extremes, novel subgroup analyses, integrative multidisciplinary omics. And you'll see as we get to the list of awardees that these features really do hold for those successful applicants. So a question that we need to ask in general, and a question we've asked all throughout this process, which has taken about a year to complete, is what is the value that we perceive at the HA to be added by CVGPS? And some of the elements of value added are listed here. The Jackson and Framingham heart studies are clearly successful cohort studies. There are large numbers of high-impact publications that have emanated from them. There are diverse scientific portfolios that have been derived from their data sets, and they have an enthusiastic and dedicated set of study participants. And we, as a facilitator, are trying to integrate those study sets and processes for analyzing the cohorts in a way that provides, if you will, a seamless platform for investigators to utilize for questions of broad relevance to cardiovascular disease. CVGPS also seeks to expand the number of investigators, study populations, and the breadth of the science that can be applied to these study sets. It really seeks to integrate new clinical and lab-based investigators who perhaps heretofore haven't had ready access to the established data sets, but with the growth in the data sets and the new omic technologies that are part of them, I think they'll offer new opportunities for people with bright new ideas and now access to the data sets they need to explore those ideas. Greater use of existing study infrastructure obviously facilitates new discovery, and the value that CVGPS adds really is in part a consequence of accommodating research beyond the parent study's original aims, and that's an important distinction to the ground rules for the original studies. So we can think of this as a population laboratory in which clinical investigators, lab-based investigators, and community-based investigators work to explore possible mechanisms of disease iteratively, if you will, feeding back information from one group of investigators to another, such that the whole is greater than the sum of its parts. That is the ultimate goal of CVGPS. Examples for use of this population laboratory are listed here. The parent study data are included in this inner box as by example, and you can see in the periphery here incorporating metabolomic analyses, epigenetic analyses, newer deep sequencing analyses, including whole genome sequencing, newer biomarkers, electronic health records, novel approaches to integrated image analyses, and unanticipated new areas of investigation, as well as linkage to other pre-existing data sets, such as molecular components of air pollution. This last slide highlights what the total vision for CVGPS is. We have a repository framework. Linked to this framework are the existing cohorts, other future cohorts, a level of partnership with the NHLBI insofar as its generous support for Framingham and Jackson over the years have led to the corpus that we have from which we're building a broader platform with their advice and consent and support, and CVGPS-based studies in particular. They all work through information and data warehouses, including some of those that are publicly available now, such as dbGaP and BioLink through the NIH, which will only lead to a greater expansion in multiple dimensions of this unique reference infrastructure. I personally think that what we can accomplish with CVGPS can be replicated in other disease domains as well, and should be broadly, nationally. And it really has been a privilege to participate in thinking through this problem, and I hope you become as excited about what it has to offer as I am as you learn more about each of the elements through the rest of our speakers this afternoon. Thanks very much, Steve.